Alright, so next we're going to talk about chromosome structure. Now, you should have already read about chromosome structure when we discussed mitosis and meiosis. Alright, so I'm going to very briefly review this topic and then we're going to move on. Alright, so here, in terms of the overall structure of the chromosome, alright, it differs between eukaryotic and prokaryotic chromosomes. Alright, so your eukaryotic chromosomes are going to be composed of DNA and proteins. Now, these proteins are histone proteins. And the DNA molecule spoils around the histone proteins. The spoil of DNA and histone proteins makes up a repetitive unit that we call the nucleosome. Alright, so the nucleosome is DNA and histones. Alright, so these histones, these nucleosome proteins, these nucleosome components, can collectively make up what we call the chromatin. Now, the chromatin can be either relaxed or it can be densely compacted. Alright, so this condensation process, alright, the condensing of the chromatin occurs during mitosis. Alright, so recall that during prophase, your chromosomes go from being relaxed in the interphase to tightly coiled in prophase. Well, that's what's taking place. The chromatin is becoming more tightly compacted. Alright, so this is generating what we visibly see as the chromosome. So eukaryotic chromosomes are linear. Now, what I mean by chromosome, when I'm talking about eukaryotic chromosomes, I'm talking about nuclear chromosomes. Alright? So we're not talking about the mitochondria, and we're not talking about the chloroplast. Alright? So we're talking about nuclear DNA. Eukaryotes, linear. Alright? Prokaryotic cells, if you recall, prokaryotic cells have typically one single chromosome that's circular. Alright, it's also double-stranded, it's also helical in nature. Right, now, this bacterial chromosome is circular. Alright, it can either be relaxed or tightly coiled. But in this case, prokaryotic chromosomes do not have histone proteins. Now, they do have histone-like proteins, but they don't have histone proteins. So this circular chromosome, it can go through this process of becoming more tightly coiled, more supercoiled, right, generating more loops. They're intertwined and intertangled. And all it takes is actually breaking one strand of a DNA apart, wrapping it around, and reconnecting it, so that you end up with a looped shaped structure like this, that's more tightly compacted and supercoiled. And here you can get an even more supercoiled circular chromosome in the bacterial cell. Now, in a prokaryotic cell, the DNA, right, is found in the nucleoid region, right? Well, the DNA is actually attached to the cell membrane of the bacterial cell. It's kind of anchored in the membrane. Alright, so this is kind of walking you through the process of going from relaxed to supercoiled DNA. Alright, so as you can see here, you have your circular relaxed chromosome on the left, and one part of the circle is going to be cut and looped around the other. As you can see here, you have a breakage. You're looping it around the other side. Alright, double strand break is resealed, and now it's more tangled. Alright, so it's very similar to an old telephone cord with all the coils. Alright, it always gets all these little kinks in it. You can never get it untangled. Well, those kinks, those tangles, represent supercoiling. Alright, so here you're actually looking at a plasmid. Alright, so a plasmid is actually, in bacterial cells, extra chromosomal DNA. Alright, so it has nothing to do with the nuclear DNA. It's completely separate. It's smaller, really, really small. Alright, and here you have a relaxed plasmid. Here you have a supercoiled. So you can tell the difference. Alright, one's relaxed, spread out. Alright, and the other is more tightly coiled and kinked and condensed. More compact. Alright, so this is showing you the genome size. Alright, so later on we'll talk about eukaryotic chromosome structure in terms of the genes that they have. Alright, compare that to prokaryotes. And how this ties in with how the cell manages to make proteins. Alright, all the little processes that are involved. Alright, but here you're just looking at genome size. And you can tell that your eukaryotic genomes are much, much larger. And they have a great range of sizes from being comparable to bacterial cells to many times larger. Alright, so looking at the human, looking at 1 to 10 to the 10th or 10 to the 11th base pairs to a little bacterium that's 1 to the 10 to the 6th base pairs. Alright, so eukaryotic genome structure is much larger. We have more genes than bacterial cells or archaea. Alright, so this is showing you, alright, for the sake of giving you an example of comparing a bacterial chromosome to, let's say, a plasmid. Alright, so the bacterial chromosome, here you're looking at E. coli. Alright, you're looking at a type strain E. coli K12, and you can see all their different genes. Alright, so here we have the origin of replication, where DNA replication starts. We have genes that are arranged into what we call operons. Now, we'll talk about operons later on in the chapter regarding regulation of gene expression. Alright, but this is kind of a preview, a snippet, that the genes are organized into these groups that we call operons, and these operons have for a particular pathway. For example, like let's say the lac operon. Right, the lac operon is involved in the degradation of lactose. All right, so lactose is milk sugar. It's a disaccharide. All right, so the genes involved in this pathway are grouped together. All right, so it's much more compact and much more organized. It's highly efficient. Okay, and then you have a plasmid. All right, many times smaller. Okay, many times smaller in comparison to the normal chromosome. And just to kind of give you a size comparison, looking at an electron micrograph, all right, all of this is the chromosomal DNA. Look at how big it is. Alright, it's spilling out of this E. coli cell. Alright, you can see these loops and coils. Alright, some of the loops and coils are tightly coiled together, others are more loose and relaxed. And then you have these little bitty coils. Alright, these are your plasmids. Now, your plasmids in bacterial cells, they encode for extra chromosomal traits, things like antibiotic resistance, resistance to toxic metals, genes involved in alternative biochemical pathways. Alright, so we know that in a molecule of DNA, you have two strands of polynucleotides. These strands are complementary to one another, and the fact that they're complementary means that they can also function as a template for the synthesis of new strands of DNA, new molecules of DNA. Alright, so Watson and Crick came up with this idea of there being a semi-conservative model for DNA replication. The idea here is that both parental strands of DNA serve as a template whereby new nucleotides are laid down, and at the end of the process, you get two new molecules of DNA. Each molecule of DNA is composed of one old parental strand and one newly synthesized strand. Now, there at the time were two other competing models, because you gotta think about it. All right, Watson and Crick discovered the molecular model of DNA. Now the race is on to figure out how you get copies of it. Alright, so they have this idea of semi-conservative model, and then other scientists have what they call a conservative model, and then a dispersive model. So now the question becomes, who is right and who is wrong? Which model is correct? So here, illustrating this semi-conservative model. So we have our parental strands of DNA, make up our DNA molecule. All right, they're held together by complementary base pairing. Now when they separate, each parental strand in blue now serves as a template for laying down new nucleotides. And now you end up with DNA molecules composed of the parent and a new strand. Parent and a new strand. So one old, one new. So this is a semi-conservative model for DNA replication. Alright, so here in this figure you're being shown the three competing models. Alright, so we just discussed the semi-conservative model. Alright, whereby your parental strand serves a template for new nucleotides to be laid down through complementary base pairing. Now the end product is that you end up with DNA that's composed of old and new strands. One being from the parent, one being newly synthesized. Now, the conservative model is whereby the parental strand, the two strands of DNA separate, okay, just as before. And they serve as a template for new nucleotides to be laid down. But what happens here is that the newly synthesized strands are released, and they come back together. They're reannealed. All right, so whereby your new strands come together and your old strands stay together and reanneal. And this keeps going on and on and on. All right, so the parental strands of DNA are never incorporated into the new daughter DNA. The dispersive model is the idea that the DNA molecule itself is fragmented, all right, and that these individual fragments serve as a template for new DNA. So the parental DNA fragments, and it serves as a template, and you end up with a mixture of old and new DNA, but in fragments and chunks in both strands. All right, so the question becomes, which model is correct? All right, which one can be backed by scientific evidence? Well, here we have a group of scientists that, through experimentation, found evidence to support the semi-conservative model. Alright, so what they did is that they labeled nucleotides of the old strands with a heavy isotope of nitrogen. And then they labeled the new nucleotides that are going to be added with a lighter isotope, so they can see where the nucleotides get incorporated. Are they going to be in both strands? Are they in one strand? Are they going to be the nucleotides that are new grouped together, nucleotides that are old grouped together? All right, so is it semi-conservative, is it conservative, or is it dispersive? All right, so this test, this experiment, should give results to indicate which is correct. All right, so here you're looking at the Michelson and Stahl experiment. All right, so what they did is they grew up E. coli in media, and in the media they had nitrogen. That was a radioactive form of nitrogen, nitrogen-15. All right, it's an isotope of naturally occurring nitrogen. All right, so they collected and they purified the DNA from the bacteria. All right, so this is generation 0. Now they took one of these bacterial cells from the original culture and they placed it in new media. And after the cells divided, they took another sample of DNA. And they took one of these progeny and they allowed the bacteria to replicate again, and they took another sample. Now these cells were cultured in media with nitrogen-14, so a lighter isotope of nitrogen. And so in this case, the DNA would be labeled with the nitrogen, whether it be radioactive or it be the normal occurring isotope. And then they took all these DNA samples and they centrifuged them. And at the end of this centrifugation process, they looked at where the DNA ended up in the tube. Alright, so here you would expect, since nitrogen-15 is a heavier isotope, it's going to be heavier and fall to the bottom of the tube, right? And if nitrogen-14 is lighter, it should be more towards the top of the centrifuge. And then if you have a mixture of nitrogen-15 and nitrogen-14, then it's going to be somewhere in between those two. Okay, so here you're looking at Michelson-Stahl's predictions for either model, for the semi-conservative, conservative, and dispersive models, and what they would expect to see from either model at the end of the experiment. Okay, so what their actual results were is after, generation 0, their starting point, all the bacteria are labeled with nitrogen 15. So it makes sense that all the DNA, both strands of DNA, have nitrogen 15 in it. Well, that works. That's correct. All right, that checks out. Now that checks out for either model. Okay. So right now we don't know anything other than what they set up in their experiment. Their initial part is correct. All right. So after the first generation. All right, so after the bacteria have replicated, they've allowed to go undergo one generation. Now, the DNA, you have one band, and this band is in between the band for nitrogen 15 and nitrogen 14. So this prediction would support either of the three outcomes. All right, because you'd expect there to be, after one generation, you'd end up with nitrogen 15, nitrogen 14 in the DNA after one generation. Now, after two generations of DNA replication, you have a mixture. All right, half of the DNA in the sample is of nitrogen 14, 15 type. The other half is nitrogen 14. All right, so that makes sense. All right, so here you end up with hybrid, or you end up with nitrogen 14, nitrogen 14. All right, so the semi-conservative model checks out. All right, so the fact that after the first round, first generation, you would have expected to see, if it was conservative, you would have seen nitrogen 14, nitrogen 15 bands, which you didn't see. You saw a hybrid. So either the semi-conservative or the dispersive model checks out after the first generation. Now, after the second generation, you would have all intermediary density types, hybrids. So all your bands would have fallen in the hybrid range, somewhere between 14 and 15. All right? But here, you actually see bands in the nitrogen 14 and bands in the hybrid. All right, so this supports the semi-conservative model for DNA replication. All right, so let's look at this again. All right, so here you're looking at all three models. All right, so here you have generation, you have one, the original DNA, you have one replication, two replications take place. So originally they're all nitrogen 15. All right, then you end up with molecules of DNA that have nitrogen 15 and nitrogen 14 in them. So one strand is composed of nitrogen 15, one strand is composed of nitrogen 14. Okay, and now the next round of DNA replication, the parental strand composed of nitrogen 15 as a template for new strands, and you get nitrogen 15, nitrogen 14 as before, nitrogen 15, 14 as before, and the nitrogen 14 strand is a template, and you get nitrogen 14 bands. All right, so again, after the first round of replication, all right, the hybrid band eliminates the conservative model. All right, so the conservative model doesn't check out. After 2 rounds of DNA replication, you end up with both the light and hybrid DNA molecules. All right, so this eliminates the dispersive model, and this supports the semi-conservative model for DNA replication.
In principle, copying DNA, a process called DNA replication, is very simple. The 2 complementary DNA strands separate, and because each nucleotide can only pair with its complement, adenine with thymine and cytosine with guanine, each strand can be used as a template to build a new complementary strand, producing 2 DNA molecules.
In the cell, DNA replication is a little more complicated, but the principle is the same. For clarity, we have untwisted the double helix. Remember that each DNA strand has a 3' and 5' end, and the strands run in opposite directions. DNA replication begins at specific sites called origins of replication. Proteins attach here and separate the DNA strands, forming replication bubbles which grow in both directions. Enzymes called DNA polymerases move along the template DNA strands and catalyze the elongation of new strands. Because DNA polymerases can only assemble new DNA in the 5' to 3' direction, only half of the new DNA can be synthesized in one continuous piece. The other half is synthesized in short pieces. As the replication bubbles grow, one daughter strand is synthesized continuously, while the other daughter strand is synthesized in pieces. The pieces are joined together by the enzyme DNA ligase. Eventually, all the replication bubbles merge, yielding 2 identical DNA molecules.
Alright, so looking at DNA replication. Alright, so DNA replication occurs at a site along the molecule of DNA called the origin of replication. Now, when you're talking about the origin of replication, bacterial chromosomes have a single origin of replication. Eukaryotic chromosomes have multiple origins of replication. Alright, so here, at your origin of replication, this is where DNA replication begins. This is where it starts. And so, DNA replication is a bidirectional process, meaning that it occurs in both directions. So the parental DNA is copied to the left and to the right of the origin. Now, your origin of replication, you get this bubble that forms when the 2 parental strands of DNA separate. You get your replication bubble. Notice that either end of the replication bubble you have this Y-shaped structure. All right, the Y-shaped structure is referred to as the replication fork. All right, later on, we're going to focus on these replication forks, and we're going to talk about what's going on at the replication fork. All right, but it's important that you note that for every replication bubble, there are 2 replication forks, one on the left side, one on the right side. Okay, and these replication forks will continue to open up until the entire molecule of DNA is replicated. All right, so that's what you see here. You see your plasmid, your bacterial plasmid, and it's undergoing DNA replication. And here you have your replication bubble that's opened up here. And on either side of the replication bubble, you have a replication fork here and a replication fork here. Alright, and the DNA will be replicated until the entire molecule is copied. And then the two molecules will separate, and you end up with two daughter molecules of DNA. Now, the same thing happens in eukaryotic DNA, except here, your replication forks, since you have, well, your origins, replication bubbles. Since you have multiple origins replication, you have multiple replication bubbles. A DNA molecule is being copied at different locations. Eventually, all these replication bubbles will merge together, and the entire molecule of DNA is copied. Alright, so here you're looking at eukaryotic DNA, and you can see that there are 3 replication bubbles in this figure. And each replication bubble has its own pair of replication forks, one on the left side and one on the right.
Replication of a DNA molecule begins at a specific sequence called an origin of replication. The two strands separate, forming a small replication bubble. DNA replication starts and then proceeds in both directions until the original DNA molecule is completely replicated. Each of the two daughter molecules consists of one old strand, shown in purple, and one new strand, shown in green. The DNA of a eukaryotic chromosome has multiple origins of replication. The multiple replication bubbles allow this very long molecule to be copied quite quickly, resulting in two daughter DNA molecules.
Alright, so here you're looking at a little bit closer what's going on at the replication fork. Alright, so we have our origin. Alright, our replication machinery comes in, separates the two parental strands of DNA. Alright, and now we have our replication bubble that opens up. And as this process is taking place, what's really going on? Alright, well, here you're looking at your replication bubble. And replication bubble, you have two replication forks. One on the left side, one on the right side. Alright, so your parental strands make up this fork. Alright, now your strands of DNA that you are about to synthesize, the new strands, alright, they are going to be synthesized in a particular manner. Alright, so again, you have to keep in mind the orientation of the molecule of DNA. Alright, so here in this figure, you're shown one strand, one parental strand, is 5 prime, the 3 prime in this direction. It's a little slight typo. And then here, 5 prime to 3 prime in this direction. Alright, so they're anti-parallel, as you would expect. Okay. Now this creates a unique situation in terms of how the strands are replicated, because the replication machinery, alright, specifically DNA polymerase, the one that's actually laying down the DNA nucleotides, is dependent upon a pre-existing 3' end to add new nucleotides onto. Okay, so let's take into account this replication fork on the left. All right, for example. All right, so DNA replication is occurring 5' and 3', correct? All right, so here, your replication machinery is adding new nucleotides in that direction. Alright, well, the replication fork is opening up to the left. Alright, so that strand of DNA is going to be replicated continuously, because as the replication fork opens, more nucleotides are going to be laid down. And it's one long continuous process. Okay, now you have another strand on the other side of the replication fork. Alright, this one over here. That is being synthesized in the opposite direction, 5' to 3'. Should actually be being synthesized in this direction, 5' to 3'. And here, for new nucleotides to be laid down, like here, here, alright, the replication machinery is dependent upon this replication fork opening up to allow new nucleotides to be added in. Alright, so this is why it's called the lagging strand, because it's having to wait for the replication fork to open up so new nucleotides can be laid down. And they're going to be added in fragments or chunks or parts. Now, looking at this figure, there's a little bit of a typo here. Alright, so your parental strands. Alright. Alright, for this to be replicated 5' to 3', that means that the parental strand has to be 5' to 3' in this direction. Alright, so the parental strand's oriented in that direction. Alright, that means that this end has to be 5', and that means this end's 3' as it is. Alright, so there's a little slight typo in the figure. Alright, but your parental strands on the outside have this orientation. Alright, so what's going on the inside in terms of how the DNA strands are being replicated is correct. Alright, the parental strands are a little off. Okay, so you'll want to make that small correction in your lecture notes. Now, one thing you'll notice that the replication forks, what's going on one side is a mirror image of what's going on the other. Alright, so listen, for imagination's sake, alright, imagine we can split this in half. Alright, on one side, the left side, for this example, alright, keep in mind this is this example. Alright, the leading strand's on the bottom. Alright, but on the other side, it's on top. Alright, same thing here. The lagging strand's on top on the left side. It's on the bottom on the right. Alright, so they're mirror images of one another. Okay. Alright, the reason being is because this strand is being replicated in this direction. Alright, so this is the daughter strand being replicated in that direction. This strand is a daughter strand being replicated right to left. Alright, now, since this side, the right side, the replication fork is opening to the right. Alright, the leading strand is continuous. Alright, so this strand here is replicated continuously. This strand here is replicated in parts because it's dependent upon this replication fork opening up to lay down new nucleotides. So it's the opposite of what you see over here. Alright, so here you're being shown a bacterial chromosome and a eukaryotic chromosome undergoing DNA replication. Alright, so again, it is a bidirectional process, whether you're talking about eukaryotic DNA, prokaryotic DNA. Alright, so these are the replication machinery, proteins, that you're going to want to be familiar with. All right, you're going to want to know what they do, what's their function, all right, and when in the process of DNA replication do they play a role. All right, so what's the order of events, and then what's the function of each of these proteins? All right, so this is a really nice little summary slide. All right, so when I go over the process of DNA replication step by step, I'm going to talk about what each of these individual molecules are doing.
The genetic information in a bacterial cell is stored in the form of a double-stranded, covalently closed circle of DNA. Replication begins at a specific site called the origin. The origin replicates, and then DNA replication proceeds in two directions. The two original strands, shown as solid lines, serve as the templates for synthesis of new strands, shown as dotted lines. This is referred to as semi-conservative replication.
All right, so looking at the initiation of DNA replication. All right, so here, one of the first things that has to take place is that your origin of replication has to be opened up. All right, now there are some other molecules that are involved that we're not going to talk about right now. They're involved in this process of opening the origin. All right, what I expect you to know is that DNA helicase comes in and it moves along the DNA in the 5' to 3' direction. And it unzips the DNA, breaking hydrogen bonds, opening up this replication fork. Now you have a part of the chromosome that's open, all right, and single-stranded. Now, how does the cell keep those single strands of parental DNA from recombining, reannealing? Well, there are single-stranded binding proteins that come in attached to the single strands of DNA to keep them from reannealing, stabilizing it, keeping the replication fork open. Now, what happens as this replication fork is opening up? Well, think about if you were to intertwine two pieces of rope, and then you take one end of that rope and you wrench it apart. Well, the other end of it, you're going to get a lot of kinks, right? Because of the torsional stress that occurs at the end. Well, this is what's happening as the replication fork is opening up, is that there is a lot of torsional stress on the DNA. It's being supercoiled, which is problematic because the DNA needs to be unwound, right, as the replication fork opens up. All right, so there is a enzyme called DNA gyrase. It's referred to as a topoisomerase. All right, its job is to relieve torsional stress in a DNA molecule ahead of the replication fork. All right, so here's your replication, your origin of replication. Here's your replication fork on either side. So this enzyme works ahead of the replication fork to relieve any torsional stress. Next, you're going to have a RNA primer that gets laid down. Now, this RNA primer, referred to as RNA primase or primase, serves as a point of attachment for new DNA nucleotides. Alright, because DNA polymerase 3 needs to have pre-existing nucleotides, a free-hanging 3' hydroxyl group, to attach DNA nucleotides to. Alright, so that's the point of the RNA primer. Alright, so that's what's going on here. Is you have your primase enzyme that's laying down your RNA primer. Now your RNA primer is laid down on the leading and the lagging strand. Now, you're going to get a single one RNA primer on your... Alright, so one RNA primer on your leading strand. Alright, because this is replicated continuously. Okay. And you're going to get numerous RNA primers on your lagging strand. Okay, because you're going to have many RNA primers that have to be laid down so DNA polymerase can lay down new DNA nucleotides. Okay, because that lagging strand is having to wait for this replication fork to open up. Alright, so this is the big picture of what's going on during DNA replication. Alright, now this slide will pop up again towards the end of this lecture on DNA replication, the mechanism behind it. Alright, so we'll talk about what's going on the leading strand, and we'll talk about what's going on the lagging strand separately. And then we'll look at the big picture again. Kind of tie it all together. Alright, so here you're looking at your nucleoside triphosphate. Alright, so here your DNA nucleotide is being attached through a hydrolysis reaction. Alright, so here two of the phosphate groups on this nucleoside triphosphate get removed, as your new nucleotide is added to a pre-existing 3' hydroxyl. Alright, now this is the job of DNA polymerase 3, is to add DNA nucleotides in a 5' to 3' direction. Alright, so this is why DNA replication, you end up with a leading and a lagging strand, because DNA polymerase can only add new DNA nucleotides in a 5' to 3' direction. Alright, so here you're looking at your parental DNA on the right, daughter DNA strand on the left. Alright, so they're antiparallel again. Notice you have a free 3' end hydroxyl on the daughter strand. You have your nucleotide triphosphate, with a little guanine attached. Here you have your condensation reaction where you're forming a new phosphodiester bond between your pre-existing daughter strand and this new nucleotide. Alright, so that's the phosphodiester linkage. That's the covalent bond.
The replication of DNA begins at a sequence of nucleotides called the origin of replication. Helicase unwinds the double-stranded DNA helix, and single-strand binding proteins react with the single-stranded regions of the DNA and stabilize it. DNA polymerase 3 is the major enzyme involved in DNA replication. DNA polymerase 3 can only add a nucleotide to the 3' end of a pre-existing chain of nucleotides, and it cannot initiate a nucleotide chain. Therefore, an RNA polymerase, called a primase, constructs an RNA primer, a sequence of about 10 nucleotides complementary to the parent DNA. DNA polymerase III can then add deoxyribonucleotides to synthesize the new complementary strand of DNA. Because the two parent strands of DNA are antiparallel, they are oriented in opposite directions and must therefore be elongated by different mechanisms. The leading strand elongates toward the replication fork by adding nucleotides continuously to its growing 3' end. In contrast, the lagging strand, which elongates away from the replication fork, is synthesized discontinuously as a series of short segments called Okazaki fragments. When the DNA polymerase III reaches the RNA primer on the lagging strand, it is replaced by DNA polymerase I, which removes the RNA and replaces it with DNA. DNA ligase then attaches and forms phosphodiester bonds. The DNA is further unwound, new primers are made, and DNA polymerase III jumps ahead to begin synthesizing another Okazaki fragment. For simplicity, DNA polymerase III has been depicted as separate units, one acting on the leading strand and the other acting on the lagging strand. The current view of DNA polymerase III is that the two subunits function together with the DNA on the lagging strand, folding to allow the dimeric DNA polymerase molecule to replicate both strands of the parental DNA duplex simultaneously. Proteins other than DNA polymerase III are not shown.
Alright, so again, reiterating this point, the leading strand is synthesized continuously, the lagging strand is synthesized discontinuously, and these fragments that we call Okazaki fragments that have to be glued back together using DNA ligase. Alright, so here, again, this is showing you a picture of your replication fork, your leading and your lagging strands. Showing you that DNA replication is a bidirectional process. Alright, so we're going to talk about the leading strand first, and then we're going to talk about the lagging strand. But again, keep in mind that these processes are occurring simultaneously.
Let's now look at the process of DNA replication more closely, starting at an origin of replication. The DNA opens up there to form a small bubble. Molecules of an enzyme called helicase attach to the DNA at the ends of the bubble and continue the unwinding of the double helix. We'll focus on one end of the bubble, on the Y-shaped region called a replication fork. The two strands would naturally tend to rewind, but are held apart by molecules of a single-strand binding protein. The synthesis of a new strand begins when an enzyme called primase attaches and synthesizes a short RNA strand that is complementary to one of the DNA strands. This piece of RNA is a primer. DNA polymerase then adds DNA nucleotides to the 3' end of the primer. It continues to lengthen the new DNA strand by adding nucleotides complementary to the template strand. Notice that DNA synthesis always proceeds in a 5' to 3' direction. The strand just made here is called the leading strand. For clarity, we'll keep the new double-stranded DNA untwisted.
Alright, so here, looking at our leading strand as being synthesized. Alright, so DNA helicase opens up the parental DNA, breaking the hydrogen bonds so that your single-stranded binding proteins can come in and attach to the single-stranded DNA to stabilize it. Your topoisomerase is going to move ahead of the replication fork, removing any form of torsional stress to the DNA molecule ahead of the replication fork. here, primase is going to lay down a RNA primer. Alright, so down here at the bottom, we have 5', 3'. Alright, this direction for our parental strand. This parental strand is going 5' to 3' in this direction. Alright, so to replicate this strand, alright, on this side of the replication bubble, alright, we're going to have a 5' RNA nucleotide, series of nucleotides that you lay down. And so at the end of this, you're left with a free hanging 3' OH group. Alright, so this is where DNA polymerase can come in and add nucleotides onto your little short RNA template. As you can see here, that's what DNA polymerase 3 is doing. Alright, so this is DNA polymerase 3. And you have this little slide clamp here. Alright, the slide clamp, alright, basically holds this enzyme in place on the parental strand of DNA molecule. Alright, so here we have our RNA primer, as depicted here. Alright, so now we can begin adding nucleotides, DNA nucleotides, that are complementary to these bases. Alright, so that is the job of DNA polymerase 3. So we would lay down adenine, guanine, adenine, cytosine, guanine, adenine, cytosine. Now your lagging strand.
The other new strand is called the lagging strand. Unlike the leading strand, the lagging strand cannot be made continuously because DNA polymerase can only add nucleotides at the 3' end. Instead, the lagging strand must be made of fragments that are linked together. Despite their names, the leading and lagging strands are actually made at the same time. For clarity, here we will just show the lagging strand being pieced together. We'll start with a brief overview of fragments being formed and linked. First, an RNA primer, shown here in red, forms the beginning of each fragment.
Alright, so we're going to ignore what's going on with the leading strand, because now that DNA polymerase is adding new nucleotides, that's a continual process until that strand of parental molecule is copied. Alright, so here we're going to focus our attention on the lagging strand. So again, the parental strand is going 5' to 3' in this direction, right? That's this guy here. Now, that means that since the replication fork is opening to the right, that our lagging strand has to be synthesized in this direction, okay, along this parental template. So it has to wait for the replication fork to open up so that new nucleotides can be added down. So again, the helicase molecule is moving in this direction. Your single-stranded binding proteins attached to the single strands of DNA. You have your little RNA primer that's laid down, synthesizes your little short RNA template. Now, you have DNA polymerase, which begins adding new DNA nucleotides to this pre-existing RNA template here. Alright, so in red, we have our new DNA nucleotides. Now it's again moving 5' to 3'. Okay. So this little fragment that's synthesized is a Okazaki fragment. And notice that now the replication fork has actually opened up a little bit more. So now we can begin replicating this region. And then we can replicate this region. Alright, so here we're having to wait for the replication fork to open up on this side so that we can lay down new nucleotides. Alright, so this is why it's called the lagging strand. Alright, so here you have your first fragment that we saw laid down. Now we have our second fragment that's laid down. So you have a new RNA primer. DNA polymerase comes in and begins adding DNA nucleotides to. And again, your replication fork is opening in this direction. So now, DNA polymerase, when it bumps into this pre-existing RNA primer, it's going to fall off. Alright? So here we have another enzyme that comes in, DNA polymerase 1. DNA polymerase 1 comes in and removes the RNA primer. Alright, it removes those and it lays down DNA nucleotides. Alright, but then we have a problem because the last DNA nucleotide that gets attached, alright, it's not going to be connected to the pre-existing DNA nucleotide chain that was synthesized by DNA polymerase 1, DNA polymerase 3, sorry. Alright, so that means we have sections of the newly synthesized strand of DNA that aren't connected. Alright, so there's a gap. Alright, so this gap has to be closed, and that's the job of DNA ligase. DNA ligase comes in and glues together this gap in the newly synthesized strand of DNA. Alright, so this gap is the actual phosphodiester bond. You have to form a phosphodiester linkage between this section here, between this section here, and this here, to make it one continuous section of DNA. Alright, so this is showing you the ligase reaction. Alright, so here you have your pre-existing DNA strand, and you have your free 3' hydroxyl. And here you have your new DNA strand. And there's a gap in place. Well, DNA ligase comes in and creates the phosphodiester linkage. Alright, so these two sections or segments of DNA can be connected. Alright, so that's what's gone on here. You have your new phosphodiester bond. Alright, so this is the big picture. Alright, we talked about what's going on with the leading strand, what's going on with the lagging strand, and that in reality, the leading strand and the lagging strand, alright, these are the newly synthesized strands of DNA that are being generated, and that the synthesis of these strands are occurring concurrently. Alright, it's a continual process. It's simultaneous.
Here you can see the simultaneous formation of the leading and lagging strands. A DNA polymerase assembles a continuous leading strand while primase, other DNA polymerases, and ligase all work together to make the lagging strand. The enzyme helicase continues to untwist the double helix, exposing more template strand DNA for replication.
Alright, so here, one of the models that you were shown in a previous video, alright, showed you this process of DNA replication occurring simultaneously between leading and lagging strands, and that your DNA polymerase 3 molecules are actually one big complex. Alright, so you have your helicase enzyme that's unzipping the DNA. They're connected to your by a connector protein to the DNA polymerase enzyme, the sliding clamp, and that basically your leading strand gets synthesized all at once, but that your lagging strand actually has to be looped out. Alright, kind of like a trombone. Your lagging strand has to be looped out, synthesized, and it comes back in. Alright, so basically your lagging strand gets synthesized in one direction, your leading strand gets synthesized on the other direction, the other side. So basically splitting, separating out these two sections of parental strands of DNA. Alright, so that one's okay, you're synthesized by the leading mechanism, the other one's being synthesized by the lagging mechanism.
Every cell in your body is produced by cell division. Before each cell divides, it must copy its genetic material in a process called DNA replication. Understanding of DNA replication comes largely from studies of E. coli, bacteria that are found by the billions in your large intestine. Let's take a look at how DNA replication occurs in an E. coli cell. As we zoom in, we see the DNA. At the origin of replication, the two strands of DNA separate, serving as templates for making new strands. The result is a replication bubble. The bubble grows in both directions, forming two replication forks. Let's zoom in on one of them. Many proteins work together at the replication fork. Only some are shown. Here, the DNA is unwound, and DNA polymerases, shown in orange, build new strands of DNA. Original parental DNA strands are shown in dark blue. Newly formed DNA strands are shown in light blue. Because strands in a DNA double helix run in opposite directions, the new strands must be made in different ways. One new strand, the leading strand, is built continuously. The other new strand, the lagging strand, is built in pieces. First, let's focus on the leading strand. DNA polymerase builds a new strand of DNA by adding DNA nucleotides one at a time. Each new nucleotide must pair up with its complementary nucleotide on the parental strand. Adding new nucleotides works the same way on both the leading and lagging strands. Each piece of the lagging strand begins with a short segment of RNA, shown in red. A clamp surrounds the RNA and attaches to DNA polymerase, which builds the rest of the new piece as DNA. When the piece is finished, it is released from DNA polymerase. how are pieces of the lagging strand joined together? A different DNA polymerase removes RNA and replaces it with DNA. However, it cannot finish connecting the pieces. An enzyme called DNA ligase joins the pieces together. Growth of the leading and lagging strands continues on both sides of the replication bubble until there are two identical DNA molecules. Although bacteria are very different from humans, the process of DNA replication in bacteria is similar to what happens in your own cells.
Alright, so what happens when this repair mechanism doesn't work as it should? Alright, so I think for every million base pairs that are laid down, you're going to have at least one single mutation in the DNA during this process of DNA replication. Now, the replication machinery also has a proofreading function, meaning that it can determine whether or not the proper nucleotide has been put in place. Alright, so there's a mismatch repair mechanism where for example, let's say you have a thymine, alright, and let's say that you have a guanine that's put in place. Alright, well that's a mismatch. Alright, the DNA polymerase enzyme can come in and replace that guanine with an adenine. Now, what happens if you have sections of the DNA, alright, nucleotides of DNA that either you have the inappropriate bases or you have some alteration, and it's a strange, rare base, right? For example, like thymine dimers. Alright, so here you have two thymine molecules, thymine bases, that actually are going to be attached together within one pre-existing strand of DNA. Alright, so this is a nucleotide dimer, alright, which has to be removed. So here you'd actually have the removal of the dimer and replace it with the appropriate bases. Alright, so here, looking at the thymine dimer, there's a light-dependent mechanism, alright, called photolyase. Photolyase comes in and will basically cleave that dimer here, so that now there's no longer a covalent bond linking those two thymine bases. The light-independent mechanism, alright, the nucleotide excision repair. Basically, these nucleotides get cleaved out. So you come in, you cut out the backbone of the DNA, and then you replace those bases with the appropriate bases, and then you remove the pre-existing segment of the DNA. Alright, so that gets removed, and now you have a gap. Alright, so your new proper bases have been laid down. This gap has to be re-annealed by ligase. Alright, so here you're being shown nucleotide excision repair. So here you have an alteration in the DNA. You have inappropriate bases that are bound together. You have a thymine dimer. Alright, so this has to be cleaved out, the backbone. That section of the DNA is cut out. DNA polymerase comes in, lays down the proper nucleotides, and a DNA ligase comes in and glues the gaps together. Now, there is a problem in that eukaryotic chromosomes, they're linear. Alright, so you can't replicate the ends of the lagging strand. Alright, that's a problem. The ends of the lagging strand aren't replicated. So that means that each round of replication, the DNA molecule itself becomes shorter and shorter and shorter. Now, this isn't a problem for prokaryotic cells because they have circular chromosomes, so everything's replicated. Alright, so this is referred to as the end of replication problem. Alright, so the leading strand gets synthesized all the way. That's not a problem. Alright, but here, looking at your lagging strand, as your lagging strand is synthesized, you'll notice that there is a section for which you have a RNA primer that's laid down, and at the end of the chromosome, at the end of the DNA molecule, you have no pre-existing RNA primer to attach nucleotides onto. Alright, so there's a section of it that's not replicated. Alright, so that means that that portion of the molecule goes unreplicated. So that means subsequent replication of this molecule of DNA will result in the molecule itself becoming shorter and shorter and shorter. So eventually, this could end up with a cell with deletions and genes that are required for the function of that cell. So more than likely that cell will probably die, alright, as a result of this. Alright, but we have telomeres. Alright, so telomeres are sequences of DNA nucleotides at the ends of the chromosome that are basically repetitive segments of nucleotides that don't encode for anything. Alright, but they help prevent this shortening of the DNA. They allow for the ends of the chromosome to be copied. Alright, so think about the production of sperm and egg. Alright, so if after each subsequent cycle, alright, cell cycle, that means that your progeny germ cells, your progeny sperm and egg cells would begin to lose genetic information, which isn't good if you're trying to pass on this genetic information to your progeny and produce a functional zygote, embryo. Alright, so we have telomerase that catalyzes the lengthening of these telomeres. Alright, so the idea here is that these telomeres are going to protect the life of the cell. And it's also going to prevent that cell from becoming cancerous. Alright, because here, after the telomeres shorten so much, alright, your cells stop dividing. Alright, so your cells have a lifespan. Alright, they age. And after so many cell divisions, they stop. Because the telomeres have shortened to the point where segments of the chromosomal DNA are being eroded. Now, your cancer cells, on the other hand, can get past this. Your cancer cells synthesize telomerase. So your cancer cells become immortalized. Alright, so here you're looking at the telomeres at the ends of the chromosome. And looking at subsequent generations of cells. Alright, so you have your first cell divides the second time, the telomeres shorten a little bit. Again, telomeres shorten a little bit. Eventually, after so many generations, your telomeres are eroded completely and the cells stop dividing. Alright, so as we age, the cells themselves, they lose chromosomes, they lose these telomeres after each subsequent cell division. Alright, so the thought is that over time, we're having more and more of these cells that are losing their telomeres. And this is tied in with the aging process. Alright, so looking at how this section at the end of the chromosome is replicated. So looking at the lagging end and looking at this missing segment of replicated DNA. Alright, so your telomerase enzyme comes in and it's going to lay down a template of nucleotides. And this template of nucleotides, as they're being laid down, so that the pre-existing DNA of the lagging strand is going to be elongated. Alright, so the parental strand's elongated, and your telomerase enzyme is going to continue to lay down new nucleotides to extend the unreplicated end. And then here, now, your RNA primer gets laid down, DNA polymerase comes in and replicates the originally unreplicated region of the DNA. And then ligase comes in and glues the gaps. Alright, so these telomeres allow for the elongation of the chromosomal DNA so that these missing regions can be replicated. Alright, so here looking at telomere length, initial telomere length, and number of cell divisions. So you can see that there's a relationship here between a positive linear relationship between the initial length of the telomeres and the number of cell divisions. Alright, so the longer the telomere length, the more cell divisions it can undergo before it stops. The shorter the telomere length, the fewer cell divisions it can undergo. Alright, so the telomere length is tied in with the basically the age of the cell. Now here, there's no real relationship between the donor age. Alright, so individuals from 0 to 25, middle-aged, late-aged, and elderly. Alright, there's no real relationship being shown here. But the cluster overall shows this positive linear relationship between the number of cell divisions and initial length of the telomeres.